We're going to stay within the Dana-Farber family for our next speaker, Dr. Rosalind Segal. Roz is a professor of neurobiology at the Harvard Medical School and Dana-Farber Cancer Institute. She became dean for graduate education in 2019. Her research group at Dana-Farber focuses on growth factor pathways critical for brain development, determining how these pathways are hijacked in brain cancers and in associated neurologic disorders. Dr. Segal is the recipient of many awards, including the NIH Director's Pioneer Award and HMS Harold Amos Faculty Award for Diversity. She's committed to training the next generation of neuroscientists and expanding diversity and inclusion at HMS. Finally, Roz is also a former runner with the Dana-Farber team. Please join me in welcoming our next speaker, Dr. Rosalind Segal.
Thank you. Well, it's great to see you all here, and it's great to represent Dana-Farber and to explain how important what you guys are doing is. The importance of being able to develop new ideas is so key to all that we do. And what I'd like to talk about today is what's really a new field, which is now called cancer neuroscience. Now, as you heard, I'm at Dana-Farber, and I have been for many years, in fact, more than I would like to count sometimes. And I've also been funded by the National Cancer Institute. So I'm really a cancer biologist. And this is an essential part of my identity. However, at the same time, I am also a member of the Department of Neurobiology at HMS, and I've been funded by NINDS, National Institute of Neurologic Disease and Stroke, and National Institute of Mental Health. For many years, this has really created a dichotomy for me, where people say, wait, are you a developmental neurobiologist or a cancer biologist? I'm like, well, I'm at the intersection of those two fields. And I have had literally people come and say, well, I didn't know that you had this whole other side of your work. And how can you spread yourself so thin? But to me, it always seemed like a very unified area where I was studying the intersection between neuroscience and cancer. And very recently, there's now a new field that's called cancer neuroscience. And so I no longer need to say I'm at the intersection of two fields. I can say I am in this new field of cancer neuroscience, which is wonderful to see. But really, it makes sense. You all know this intuitively. We know that the mind affects cancer risk. Thinking about friends, loved ones who've had cancer, how many times do you say, gosh, it just seems so odd that this came on after so much stress in their life? I wonder whether these worries, these issues, these concerns led to the cancer in some way. And in fact, we probably don't know, but it is clear that in fact, the stress can modulate, if you will, the cancer risk and just push it upward. How does it do this? It's because of the nervous system. As we think and we get tense and we release neurotransmitters and we have more electrical activity and more anxiety, what happens? There are changes in the neurons in your body. And these in turn affect tiny tumor cells that otherwise you might eradicate. And the neurons impinge on the tumor and its microenvironment to enhance that tumorigenesis. And this is part of the field of cancer neuroscience. There are so many ways in which neurons affect cancers and cancers affect neurons. So one thing, as I just mentioned, is that when you're stressed and you release norepinephrine, that is something that many tumor cells respond to. And how do they respond? Many of them respond by growing, dividing, proliferating, all the things we don't want cancer to do. And so that stressful response and that autonomic system is causing an enhanced risk of the cancer growing and becoming a real issue. In other ways, neurons can affect the interactions among tumor cells and so cause them to help one another grow. Neurons also, because they have these very, very long axons that can be a path, say, from your toe up to your back, these are great highways for tumor cells to crawl along as they metastasize from a site of origin to other places in the body and so become more malignant. We know that not only do neurons affect tumor cells, but also tumor cells affect neurons. Tumors make a lot of factors, including one that I'm going to be talking about most, which is called nerve growth factor, NGF. And these factors contribute to the growth of the neuron into the tumor. Why are cancers so painful? It's not the cancer that's causing the pain, it's your nerves that are causing the pain. Why are your nerves causing pain? Because the tumors have made the neurons grow into them and sense that the growth and the stretching of the tissues from the tumor. And so the tumor is causing pain by enhancing the growth of these neurons that are nearby. The tumors are influencing factors in the environment and so changing the nearby neurons. Tumors can also actually injure the neurons and cause other sequelae for patients. So neurons are affecting cancers and cancers are affecting neurons. So it makes sense that there's a field that includes the two of them. Not only do cancers and nerves affect one another, but the treatments that we use for cancers affect the cancers, but also affect neurons in many ways. So although we're hoping that the chemotherapy this patient is getting is going to kill her cancer, it can also cause changes in the brain. Chemobrain, chemofog, or to use the more technical term, CICI, chemo-induced cognitive impairment, a real side effect of the drugs that are effectively used for cancers. Many cancer drugs cause hearing loss. This is particularly true for some of the drugs that are used for kids. And this is something where you really don't want them to be deaf. So how do we effectively treat them without having these deleterious effects? And one that was the subject of one of my bar awards, which was when we started working on CIPN, chemotherapy-induced peripheral neuropathy, which was a new idea that I had many years ago. So I asked the bar for funding for this and started to work on this work that's now gone much farther and is now funded by NIH. But more than 50% of patients treated with chemotherapies develop chemo-induced neuropathy, where they have pain, numbness, tingling in their hands and feet. I became interested in it after my sister was treated for breast cancer and developed this. But this is seen in probably over half of our patients, and we currently have no treatments for it, 0. So how do we develop treatments that will prevent these effects, but still allow us to treat the cancer? And this is something that we're actively working on. And this is the kind of work that your runs and your fundraising is going to support. When I think about grit and gumption, the person I think of is the person who really, in my mind, was the first cancer neuroscientist, Rita Levi-Montalcini. And this is a picture of her here. Rita Levi-Montalcini was, in the 1930s, way ahead of her time, a faculty member in science at the University of Milan. I think she was one of the only women in her department. But so she was on the faculty in Milan in 1939, and as you may be able to tell from her name, she was a Jewish-Italian scientist. And so when Mussolini took over, and the Jews were all fired from the universities, she and her family left Milan because they were in danger, and they moved to a very small cottage in the Alps. When she moved to the cottage, she took her microscopes with her. And so in this cottage, which basically was a one-room cottage, there she was with her brother, his kids, and her mother. And she went around to the neighboring farmers and asked for eggs to feed the children. Sometimes they gave them to her. And so she took the eggs home, and what did she do? She did an experiment with them. What she was working on was trying to understand the nerves that go to the limbs, the wings, and the feet of the chicks. And she was a developmental neuroscientist. And so she was trying to understand how the nerves go to the right place. And what she found was that if you pluck off one of the limbs of the developing egg, the developing chick inside the egg, there are fewer nerves going to that area. And if she took the wing of one chick and put it on another chick, then there were more nerves that innervated this new wing. And she said, oh, there must be something in the wings that causes these neurons to grow out. And she called it nerve growth factor. She hadn't purified it or anything yet. But at the same time, and so she very carefully made drawings of her results, which showed this outgrowth of the nerves and showed that the limbs were doing something to cause this. She made the pictures, and then she took the rest of the chick and she cooked it and fed it to the kids. Because they were hungry. So after the war, the Rita Levi-Montalcini and her family came back down from the Alps, and she was reinstated in her position at Milan. And she published the work that she had done, showing the pictures that she had drawn of these chicks. Now, it turned out that a group at Washington University in St. Louis, Victor Hamburg, was working on a very similar problem. And what Victor thought was that the limb was releasing a factor that caused the nerve cells to proliferate. Whereas Rita thought what it was doing was causing them to survive and grow out. So when she published her work, which directly contradicted his, what was going to happen? Well, what happened was that he invited her to come to Washington University and to work with him to figure out the answer. And so she did, and she worked there for more than 10 years. And the two of them together brought in another person named Stanley Cohen. And they realized that, in fact, this factor, this nerve growth factor, was causing outgrowth of the nerves and causing more of them to survive. And they wanted to purify it. And then, in what makes her the first cancer neuroscientist, she said, well, where am I going to get enough of that factor? Because there's not that much in the chick limb. And she realized that when she took some tumor cells that were growing in the lab, sarcoma cells, S, and she put them next to the nerves, it caused this beautiful outgrowth, what you can see here, of the axons, and they just grew right to the sarcoma. And she said, oh, these cancers, they make the same factor as the limbs. That makes sense, because this was a sarcoma that came from actually a limb. And so they used the sarcoma to purify nerve growth factor, the work that led to the Nobel Prize for her. Now, when I started my lab many years ago, the first thing that I was working on is how these tumor cells signal to the neurons to tell them to grow their axons and for the neurons to survive. And how does that work? What's that process? Is this something we can figure out? Which it turned out to be. What I learned from Rita Levi-Montalcini is how important those interactions between cancer and the nerves are, and how we need to understand them. She also clearly epitomized a personal story of grit and a history of working together with others. So cancer neuroscience is now considered a field. This was the first meeting of the cancer neuroscientists at Cold Spring Harbor, and I was fortunate to be one of those who was there. And it is now considered a new area to be explored and to use to develop new therapies and new treatments. It involves the neural cancer interactions, where neurons release neurotransmitters that affect cancer cells releasing NGF and other molecules that affect nerves, and nerves in turn causing effects on the cancer cells. And then the ways in which stress causes a change in predisposition to cancer. And finally, the way in which cancer therapies not only do what we want them to do, which is to kill the cancer, but often, unfortunately, have these unintended side effects. And so it's a really exciting area to be in. And with the help of all of you and of all the runners and of all the great support that Dana-Farber has, there is a lot more to come. And this is my group currently of cancer neuroscientists and previous groups that have been around, including Cahal's drawings. And I want to thank you all for being here and for all the ways in which you support the scientists here at Dana-Farber who are doing really innovative and important studies to try to improve life for all those with cancer. So thank you.